A giant rat can diagnose tuberculosis faster than $1 million laboratory. And the reason is written in its genes. While humans evolved massive brains to build hospitals, we traded away something essential. The hyperspecialized olfactory receptors that once let our ancestors smell death itself. African giant pouched rats. Honeybees. Dogs. They can detect lung cancer, tuberculosis, malaria, even COVID by scent alone. Long before any blood test comes back positive. This is not alternative medicine. This is peer-reviewed science. And it is forcing us to rethink everything we know about diagnosis. In Tanzania, an organization called APOPO has trained African giant pouched rats to do something extraordinary. They screen sputum samples for tuberculosis. One rat can process 100 samples in 20 minutes. A human lab technician might process 20 samples in that time. The rat? 5 times faster. By the end of 2022, these rats had screened over 870,000 samples and found more than 26,000 tuberculosis patients that conventional tests had missed. Published studies show the rats achieve 99.15% sensitivity, 92.23% specificity, 93.14% overall accuracy. These are not random guesses. These are highly reliable diagnostic results. The rats detect Mycobacterium tuberculosis by smelling volatile organic compounds, VOCs, that the bacteria release. Specific chemical signatures that human noses are simply not equipped to perceive.
But rats are not the only animals outperforming our technology. In 2024, researchers at Michigan State University published a study that stopped the medical world cold. They harnessed honeybees in 3D printed plastic harnesses, performed brain surgery to attach electrodes, then passed air containing lung cancer biomarkers over the bees' antennae. The bees' brains fired distinct neural patterns for cancer breath versus healthy breath. The system classified lung cancer with 93% accuracy. At concentration levels of parts per billion, parts per trillion. The honeybee olfactory system can detect minute chemical differences between types of lung cancer. Small cell versus non-small cell. This is a living sensor that outperforms many electronic noses.
So how do they do it? The answer lies in genetics. Specifically, in the olfactory receptor genes. In 2020, scientists sequenced the olfactory receptor repertoire of the African giant pouched rat. What they found explains everything. Humans have roughly 5 million olfactory receptor cells. Dogs have over 200 million. Rats have a similarly massive repertoire. But it is not just about quantity. It is about the specific protein structures encoded in their genomes. These hyperspecialized olfactory receptor proteins have binding pockets shaped to capture specific volatile organic compounds. When a TB bacterium releases methyl nicotinate, the rat's olfactory receptor binds it with precision. This triggers a neural signal that the rat's brain recognizes as tuberculosis positive.
Which brings us to the uncomfortable question. Why can't we do this? Why did humans lose this ability? Evolution made a trade-off. As our neocortex expanded, as we developed language and abstract reasoning, our olfactory machinery atrophied. Of the roughly 800 olfactory receptor genes in the human genome, about 60% are pseudogenes. Non-functional. Broken. In rats, the vast majority are intact and active. We literally lost the genetic instructions for building these receptors. Early humans relied on smell for survival. But as we developed social structures, language, and medicine, the evolutionary pressure to maintain acute olfaction diminished. We traded raw sensory ability for cognitive processing. We cannot smell disease. But we can build a microscope. Sequence a genome. Invent a vaccine. The trade-off gave us civilization. It also left us chemically blind.
Researchers are now trying to reverse this. If animals can detect disease by smell, can we build technology that does the same? Electronic noses exist. They use arrays of chemical sensors to detect VOCs. But they struggle with real-world environments. The honeybee research points to a different approach. Instead of building artificial sensors, why not interface directly with biological ones? Scientists envision a future where a patient breathes into a device, and inside that device, living honeybee neurons analyze the breath in real time. A biological sensor grown in a lab. In Tanzania, APOPO's rats are already doing this at scale. No machine. No microscope. Just a rat and its genes.
So here is the uncomfortable truth. A rat that costs a few dollars to train can detect a disease that $1 million laboratory sometimes misses. And the reason is not magic. It is genetics. Deep within their genomes, these animals carry protein structures that we lost millions of years ago. They smell what we cannot. They detect what we miss. And in doing so, they remind us that evolution does not produce better or worse. It produces trade-offs.
If this changed how you see animals, hit subscribe. We explore the hidden science of the natural world every week. Comment animal and I will send you the full list of studies referenced in this video. Next week, the animals that can detect earthquakes before they happen. The frontier moves fast. Keep up.